Now let's dive into some details about prokaryotic. Prokaryotic gene regulation. So just as an overview, before we get into a specific scenario, generally, gene expression is regulated at the level of transcription. We're going to find that in eukaryotic systems, we have transcription regulation, but there's regulation of gene expression at a number of different levels in the process going from the gene to the functional protein. So, but in prokaryotes, generally, if a gene is to be regulated, it's regulated at the level of transcription. And often, genes are negatively regulated. So meaning repressor proteins, when bound to the DNA, kind of silence the expression or downregulate expression of the genes. To get them transcribed, you have to get the regulator protein to dissociate from the DNA. So negative regulation is common in prokaryotes. We'll find out in eukaryotes, it tends to be positive regulation. Doesn't mean it's the only thing, but it means it's commonly negative regulation is the system used. And many genes in prokaryotes are coordinated in their regulation. So many genes have or demonstrate coordinated regulation. So what does that mean? In coordinated regulation, it's where a group of genes So a group of genes, more than one gene, and often these are genes that code for proteins involved in the same biochemical pathway. So in coordinated regulation, there's a group of genes, often with products involved in the same biochemical pathway, that are transcribed and translated together. It's kind of a really beautiful, efficient sort of situation, right? Because if you're breaking down, you need to break down some macromolecule, and maybe there are 2, 3, 4, 5 genes involved in that, or proteins involved in that pathway, then why not make them all together? It's kind of like a one-stop shop. So we call genes, and so if you think about this, if we've got DNA, here's our level of DNA. So in coordinated regulation, you might have gene A, gene B, gene C, and they're all under the control of the same promoter. So they're transcribed, all 3 of them are transcribed together, and then they're translated together. We call this system where we have genes under the control of the same promoter, this is called an operon. And this is specific to prokaryotic. We don't see operons in eukaryotic systems. So let's take a look here. Again, kind of just demonstrating here. We've got our promoter region. Any kind of regulatory regions might be around here as well. Gene A, gene B, gene C. Now let's look what happens when here's our, so the result, the product of transcription of coordinated genes, product of operon transcription, is that we have a messenger RNA that codes for a number of proteins. And we call this polycistronic mRNA. So for coordinated gene regulation, when they're transcribed, the result is that you have many cistrons, meaning coding for different genes, messenger RNA. And look what happens here. So we had gene A, gene B, gene C, they all get transcribed together. So this message contains information to make 3 different proteins. Each one of those genes has its own unique start and stop codon. So the ribosome comes on and starts translation of the opener reading frame. We get protein A made, and then here the ribosome can stay on here, or a new one can pop on and start translating it again, protein B, and then protein C. So let's take a look at a very specific operon system, and we're going to take a deep dive into this because I feel like if you fully understand how this works, you understand prokaryotic regulation of gene transcription, and you can make any kind of modification to imagine other situations for regulation. So we are going to use the lac operon. So it's an operon system, and it's called the lac operon. It is an inducible system. Coordinated with 3 different genes whose products, so the products of these genes, or the proteins they code for, are involved in the metabolism of lactose. Now, lactose should ring a bell. It's a sugar, it's milk sugar, right? So the lac operon codes for genes that are involved in lactose metabolism. So let's take a look at lactose metabolism. So lactose is a disaccharide. It's a disaccharide of glucose and galactose. So it's a disaccharide of glucose and galactose held together by a glycosidic linkage. Now, you remember from cell bio, this does not enter into glycolysis. You have to break this bond to get monosaccharides that can then enter glycolysis. Glucose enters directly, galactose can be modified and then enter glycolysis. So used for energy to generate ATP, right? But you have to break this bond first to be able to utilize lactose. So lactose, the disaccharide, can be broken down into glucose and galactose. And that's breaking a covalent bond. That means there's an enzyme involved. The enzyme that catalyzes the breaking of this glycosidic linkage is called beta galactosidase. Shortened beta-gal. So beta-gal is the enzyme that breaks it down. So it's a pretty simple kind of metabolic pathway, right? You've got one enzyme, it breaks the disaccharide into 2 monosaccharides, and then those feed into glycolysis for energy utilization. So let's take a deep dive into, oh, and here's just another, it also does sort of this isomerase reaction. We're not going to worry so much about this. This is lactose being converted to allolactose. We're not going to so much worry about that, but just recognize that there are multiple pathways that can occur in lactose metabolism. Let's take a deep dive into the operon. So the components of the operon and then how the operon transcription is regulated. So the genes that are coordinated together under the control of the same promoter are called structural genes. Now, it's not like a physical scaffold. It's not, doesn't have anything to do with actual structure, but it's how we refer to the genes that are under the control of the same promoter region. So the structural genes, there are 3 of them. There is the gene LAC-Z. LAC-Z, here you see it's 3,072 base pairs. Genes are just recipes. It codes for beta-gal, right? So the beta-gal that we just mentioned that is involved in breaking down lactose. The next one is LAC-Y. And LAC-Y codes for an enzyme called lactose permease. So permease is a membrane pore, it's a transmembrane pore forming protein. and it's selective. So here's a bacterial cell, here's permease embedded in the membrane. So what it allows it allows efficient uptake of lactose. Some lactose can get in through glucose transporters and others, but it's a pore forming protein that allows a much greater increase in lactose uptake from the cellular environment. Our third and smallest gene is LAC-A. It codes for a protein called transacetylase. Its role is a little less clear or obvious, but it's thought to be involved in sort of detoxification of byproducts from lactose breakdown. And we're going to leave LAC-A out of our consideration now, but it is one of the structural genes that is involved here. So we've got our three genes that are coordinated. They're going to be transcribed and translated together. But we also have our DNA regions involved. So the regions that are involved, so I'll just blank out this screen. So DNA regions outside of these structural genes that are involved. Well, you know, to have transcription, what must you absolutely have? The promoter region. So we'll call that the P region. In the promoter, as we've learned about, is the RNA polymerase binding site. It is required for transcription, required. If you do not have a promoter, you do not have transcription. There is also an operator, which is actually overlapping the promoter region. They're not mutually exclusive. It sort of overlaps the promoter region. So here's our both our promoter and our operator binding region in here. And remember what an operator is? This is the repressor binding site. When a repressor is bound, to this operator, it blocks transcription by forming a repressor loop. So let's take a closer look at this, at the mechanism of this. Here's our promoter sequence in orange. Remember our -10, our -35 sites. This operator actually overlaps this. There's another little region over here that's not shown. And it goes into the gene itself a little bit. There's the operator binding site. Here is the repressor protein. Remember, it's a DNA binding protein. It recognizes this sequence. So look at these. These are both considered the operator binding site in yellow. So it's sequence that's recognized and bound by the repressor protein. And when it binds, it causes this conformational sort of tweaking of the promoter region, and polymerase can't bind to this. It's a repressor loop. So those are the major DNA regions involved. And there are also some other things at play. In addition to the promoter, the operator, the structural genes, there are other things involved. There's the repressor protein itself. Where does the repressor protein come from? There has to be a gene that codes for the repressor protein, and that is the LAC-I gene. So this is the gene that codes for the repressor protein. It is not under the control of the same promoter. It has its own promoter region, and it is constitutively expressed. That means if you bust open a bacterial system, there is going to be repressor protein floating around. This is not regulated, it is constitutively expressed. And then the final thing that we have to consider is our environmental factor that is going to regulate all of these players, and that is lactose. Lactose is a sugar, and it itself, the thing that is going to be broken down, acts as an inducer. And remember what inducers do, they bind to repressor. So it's an inducer that binds to the lac repressor. It causes it to dissociate from the DNA. So when the repressor is gone, then this repressor loop is gone, and it allows transcription. So this is describing the negative regulation of the lac operon. So let's just kind of take an overview of it. So let's say the negative regulation of the lac operon, because we'll find out there's also some positive regulation, but we're going to nail down negative regulation first, where lactose acts as an inducer. So in conditions of low lactose, let's consider what's going on. In conditions of low lactose inside of our bacterial cell, we have our bacterial operon, our lactose operon. With low lactose, the repressor protein, remember, is constitutive. The repressor binds the lac operator, forming the repressor loop, and transcription is off. So here we have our repressor protein being made by the LAC-I gene, the recipe for it, bound to the operator, blocking transcription. Now, what happens when our little inducer molecule, when environmental conditions change and lactose is present in the environment? So when lactose concentrations increase in the environment, small amounts will diffuse into the cell originally. And it will bind to the repressor. That's the repressor protein. Let's go to the next one. Here we've got it binding to the repressor protein and causes the repressor to dissociate. Now, once the repressor is out of the way, RNA polymerase binds and transcription occurs. Now this is very elegant. So just let's stop and think about that for a second. The genes that are under the control of this repressor system and have now just been turned on, they've been turned on by the presence of lactose, and then they're going to make proteins that are involved in actually metabolizing lactose. Think about how energetically brilliant that is. You don't want to transcribe any kind of synthesis pathway is going to utilize energy. So there's energy consumption in transcription and translation. Why would you waste a cell's energy making proteins that you don't need? When do you need it? You need it when lactose is present. And it is actually lactose itself that causes the production, the synthesis of those proteins that are then going to be involved in utilizing lactose for an energy source. Brilliant, brilliant system. And this system was determined, the role of the operator, repressor, all of those things were determined using different bacterial mutants. And so I'm going to introduce, well, let's watch the movie first and then we'll look at the mutants.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
So this system, as I mentioned, was discovered by studying mutations to this operon system and then seeing what the outcome was. So mutations to the operon and how they affect transcription. And so I feel like if you understand and you can predict what these mutations would do to the operon system, it's going to increase the comprehension of this system in whole. So let's talk about the different types of mutations to the operon and what the expected outcome would be. And let's imagine the operon like this. We've got the LAC-I gene is upstream of the promoter region with the operator kind of overlapping the promoter. Then we have our LAC-Z gene, our LAC-Y gene, our LAC-A gene that we're not going to consider, but here's our promoter operator. The LAC-I gene is coding for the repressor. So let's actually just do this. Here's the repressor protein with its DNA binding site combined to the operator or not. Okay, so let's imagine now. One type of mutation is a promoter mutation, a P- mutation. This is a null mutation in the promoter. So the promoter sequence is mutated and RNA polymerase cannot bind. What is that going to do to transcription? Well, it's required for transcription. So this P- mutation would result in no transcription. Another type of mutant was called the OC. This little C stands for constitutive, and it is a mutation in the operator region. So the operator, it's an operator mutation in the sequence, and that doesn't allow repressor to bind. Now let's think about that. In conditions where there is no lactose present, the repressor normally is bound. But here it's a mutation in the operator and the repressor can't recognize it and bind to it. So what is that going to do? It's going to allow for transcription even in the absence of lactose. That means that the expression, the transcription of these is going to be constitutive. So this stands for constitutive. Remember what that means. So this is a constitutive mutation. This mutation that doesn't allow repressor to bind allowed the genes to be transcribed and expressed all the time. So that suggested that this was, in its normal state, was involved in a repressor type system. There's also an I- mutant. The I- mutant, it's a null mutation. It's a null repressor mutation. That means you have no functional repressor. This is out of the system. So if you have no functional repressor, so no repressor binds, no binding to the operator, the result of that is going to also be that transcription is going to be on whether or not you have lactose. A different mutation in the repressor gene, the LAC-I gene, is called the I superscript S. This is the super repressor. This is a mutation, so it's the super repressor mutation. And it's a mutation in the gene that results in a mutation in the LAC binding site. So it's a mutation that does not allow lactose to bind. But it still has a functional DNA binding site. So actually, if let's kind of look at it just like this. So in a normal wild type, in the wild type repressor, there's both a lactose binding site and a DNA binding site. In the super repressor, the IS mutation, this is blocked, but this is not. So that means that the repressor is always bound to the operator. That means that polymerase is never going to get in there. And so therefore, you have no transcription, whether or not you have lactose. And then we can have mutations to the structural genes themselves. So let me clear this out. Do this, make my eye cozy. You can have mutations to these genes themselves. And a Z- mutation is a null mutation. It's a null mutant where you get no functional beta gal. And we can also have not a P, but a Y- mutation. That's a null mutant that results in no functional permease. So these are different mutations to the operon, which helped scientists to understand the full functionality in this operon system, what all the different components do. One last sort of dive into the LAC operon is that the major mechanism of regulation of gene expression for this LAC operon is the negative regulation by the repressor protein. But there's also positive regulation that can occur, that does occur. And so let's touch on the positive regulation of the LAC operon. In positive regulation, there is a cyclic AMP, and I'm just looking at this AMP, so this is like a modified nucleotide, cyclic AMP cap complex that acts as an activator. That acts as an activator for transcription of the lac operon genes. The cyclic AMP cap complex is regulated by levels of glucose. So it's regulated by glucose concentrations in the cell. So let's take a situation where we have low glucose concentrations. When a cell has low glucose concentrations, think in terms of energy. Low glucose means that the cell starts to become energetically depleted. ATP levels go up. When ATP, the energy levels, go down, and when those levels go down, this modified version of ATP, cyclic AMP, increases in concentration. There's an enzyme that's activated, but so it's a inversely proportional relationship. So when glucose goes down, energy levels go down, but cyclic AMP levels go up. This is sort of indicating that the cell's in a low energy state. And it acts as sort of an effector molecule here. And it binds to, this cap protein. So CAMP levels, when they go up, it binds to the CAP, which stands for catabolite activator protein. When cyclic AMP binds to CAP, it acts as an effector molecule and it causes it to bind to the activator binding site. And it recruits RNA polymerase to the promoter region. So when glucose levels are low, this acts as a positive regulation system, bringing in RNA polymerase at a greater rate than it would otherwise. So in conditions of low glucose, we have an increase in transcription. Now, if there is no lactose around, here's where the repressor binds, and it's going to override that positive regulation. So that's why we say that this system is primarily regulated by the negative regulation of the repressor system. But when this is off the operator, then this low glucose conditions are going to recruit in polymerase and cause an upregulation in transcription. When we have high levels of glucose, the cell is happy. This is fed. That's a well-fed cell, lots of sugar, and it's very happy. It's clicking through glycolysis, making all of its ATP. The ATP levels are high, and the cyclic AMP levels decrease. The cell is happy. It's fed. So what happens here is that no complex forms. Therefore, it does not bind the activator binding site. So polymerase can get in here, but it doesn't come to the promoter region as efficiently. So we have limited transcription because polymerase doesn't get in here quite as easily. And if you think about it, you don't need to metabolize lactose if you have glucose. And so both the negative and the positive regulation systems are in play with the lac operon. So let's think about both glucose and lactose concentrations and how that is causes a sort of coordinated regulation. We can think about it at both the molecular level, but also kind of back off the details and think about a big picture. Think about, imagine yourself as a bacteria, and if you're swimming around in, a nice rich media of glucose versus not, and whether or not you have lactose. So let's take this situation where we have, let's go high lactose and high glucose. Let's think about what's on the operon system. So here's our IPOSY. So in the case of high lactose, would the repressor bound? So no, there's no repressor on the operator. And we have high glucose. So that means we have low cyclic AMP. So we have no repressor bound and we have no activator bound. We'll get transcription every once in a while if polymerase comes in here and hits and does a transcription, but that's going to be limited. So we would have limited transcription under this condition. What about if we had low lactose and high glucose? What would be different about this system? Well, in low lactose, high glucose, let me draw out my IPOSY again. Let's think about it. Low lactose, is the repressor bound? Yes. The repressor is bound and it's blocking polymerase. You can bring in all the polymerase you want. You wouldn't though, because there is no activator. And the repressor is bound. In this case, you get no transcription. Whether or not no transcription. What else do we have? How about a high lactose, low glucose condition? High lactose, do we have repressor bound? Nope, repressor is not going to be bound. So no repressor. What about with low glucose? Well, that means cyclic AMP levels are going to go up. And so in addition to this, we would have activator bound. So we would have that activator complex bound. It's going to recruit in polymerase, and we would here have optimum transcription. And I guess we have one final situation. Which one are we missing? Oh, high lactose and high. No, get that one. What are we missing? Oh, low lactose and low glucose. Let's see. Let me go ahead and just erase one of these and do a new one. Okay, so in conditions of low lactose, what happens? We would have repressor bound. So we know we're getting nothing. We might be able to bring in the little activator and it might be recruiting it, but it can't go anywhere because there's a repressor loop. So you would have no transcription. And this is the coordinated regulation of the lac operon.